first one is that there are a great variety of possible pathogens. So, as you may have known, there are many types of bacteria which may cause disease in the gastrointestinal tract, right? And this makes it challenging for us to identify any pathogen or to identify the pathogen or the microbe responsible for the problems in the patient, right? The second problem is the what? Is the normal flora. As you know, the flora is the part of the body containing the highest concentration of normal flora, right? And so this makes it challenging for us. It makes it harder for us to identify which of these microbes is responsible for the disease. If any microbe is indeed responsible for the disease, right? Maybe it is another microbial disease. So this will create problems for us also. Now, in order to face these challenges, we adopt the following measures. The first one is arranging bacterial GI pathogens in order of priority, right? So we are unable to identify if a GI bacterial pathogen is responsible from the first instance, right? Because there are many different bacteria, so we have to put what? We have to arrange our priorities. We have to look for the most important bacteria. If we are successful, that's it. If not, then going down the list, right? So, prioritizing. The second is use of media with very selective properties. Now, as you know, selective media are those media which permit the growth of some types of bacteria but inhibit the growth of others, right? So, selective media makes it easier for us to identify or to filter out most of the normal flora, right? So, what will remain most likely, or we hope that the bacteria that remain and that are able to grow in the media are those that are pathogenic, that are more pathogenic. Understand? Now the difference between selective and differential is that selective medium, it permits the growth of some bacteria and inhibits the growth of others. Differential, it allows the growth of different types of bacteria, but it gives different properties to what? To different types of bacteria according to their characteristics. Now, bacteria routinely look for that practice are what? Shigella, Salmonella, and Campylobacter jejuni. So, of course, it depends on the lab, right? So, different labs have different routine bacteria to look for, right? But most labs, they prefer to look for these three. Some concentrate on Shigella and Salmonella only, and some may increase the list, right? But let us concentrate on these three. Maybe on Shigella and Salmonella. After that, we talk a little about Campylobacter jejuni and Vibrio cholerae. Now, other types of bacteria may be looked for upon clinical or lab suspicion. Clinical or lab suspicion. It means if the doctor feels that maybe a certain type of pathogen or a certain type of bacteria is responsible for causing disease of THEC, enterohemorrhagic Escherichia coli, or 157H7, right? Or that this is a case of cholera, for example. Then the doctor must put his suspicion clearly in the lab test requisition form. Of course, there is Yersinia enterocolitica and Clostridium difficile. Clostridium difficile is a major cause of hospital-acquired diarrhea. It is an anaerobic bacteria. We will not talk a lot about these two, right? We will talk more about these five. Now, major properties of Enterobacteria. Enterobacteria here, it means what? Members of the family Enterobacteriaceae. Enterobacteriaceae are a family of oxidase-negative, gram-negative bacilli, right, which are glucose fermenters, and they have certain other characteristics. Now, these Enterobacteriaceae or Enterobacteria, there are many different genera, and these bacteria, they affect different parts of the body. Now, of course, here we have Shigella and Salmonella, which are major GI pathogens. Also, E. coli. Some types of E. coli, we call them, some pathotypes of E. coli are able to cause GI disease. Enterotoxoidemic Escherichia coli, enterohemorrhagic, etc. Right? Now, Klebsiella pneumoniae and Proteus, they are not causes of GI disease, but the problem is that they may be present as normal flora. Therefore, we must be able to what? To differentiate or tell them from the other members. Now, starting with lactose fermentation. E. coli is a very strong lactose fermenter. Klebsiella pneumoniae is a lactose fermenter, but not as strong as E. coli. Shigella, Salmonella, and Proteus, they do not ferment lactose, right? Now, hydrogen sulfide, Salmonella, and Proteus produce hydrogen sulfide. The other three, they do not produce hydrogen sulfide. Now, these are important because when talking about media, we will learn how to differentiate these bacteria from each other. Now, of course, you know what is ended. Indole, methylred, Voges Postcode, and citrate, right? We're supposed to learn this in your first clinical microbiology course. Now, ended, indole, methylred, Voges Postcode, citrate. E. coli plus plus minus minus, meaning that indole positive, methylred positive, Voges Postcode negative, and citrate negative. Shigella, here V, it means variable, right? It means sometimes positive, sometimes negative, according to the exact species. But the methylred is positive, negative, negative, right? Salmonella, negative, positive, negative V. Klebsiella pneumoniae, negative, positive, and Proteus, it is positive for methylred, but the others are what? Variable, depending on the species. So some species may be citrate positive, citrate negative, etc. Right? So remember the ones that are clearly positive or negative and ignore the rest.
Okay.
Urease, Klebsiella pneumoniae, and Proteus. Proteus, it is a very strong urease producing bacteria, right? Urease. Other properties, Shigella is immotile, it cannot move. Klebsiella pneumoniae, it produces mucoid colonies and strong gas production, sometimes leading to the formation of bubbles or detachment of the media, especially if they are in tubes. And Proteus, swarming motion. Swarming motion, it means it produces a wave-like movement on top of the what? On top of the media. We call this swarming, right? It expands from the colonies to the outside because it is hydromophile. And so it will create a wave-like what? Appearance on top of the collagen. Now, selective media used for GI specimens. Selective media used for GI specimens. Three types of media in general are used for culturing GI specimens. Now, enrichment broths, selective media, and the third one is highly selective media. So enrichment broth. What is an enrichment broth? Broth, it is a liquid medium. We call a liquid medium what? A broth. Right? Now, they are especially useful when looking for Salmonella species. From their name, they are called enrichment. Why? Because they allow us to what? To increase the number of suspected pathogens. Right? So, when we put the stool sample, if we suspect that this patient has Salmonella infection, then it is advised to put it first in this enrichment broth, and after some hours to culture from the broth onto an agar, right? In order to give more chance for any Salmonella that may be present in the specimen to what? To increase in number, right? Therefore, we call it enrichment. Now, some types of enrichment broths are selenite broth and gram-negative broth. Selective media for isolating gram-negative bacteria and identifying lactose fermentation. So selective media are not only selective, but are also what? They are also differential. So they are selected for gram-negative bacteria against gram-positive bacteria. So they do not allow gram-positive bacteria to grow. Therefore, they are what? Selective. They are differential because they help us in telling those lactose fermenting bacteria from lactose non-fermenters. Right? So, selective and differential. They include McConkey agar and eosinophilin blue agar, EMB. Right? EMB agar and McConkey agar. These are both selective media. Then we have what we call highly selective media. Highly selective media. Now, from their name, they are not only selective against gram-positive bacteria, but they also select against many gram-negative bacteria. Right? So they help us in isolating more and more those two genera, Shigella species and Salmonella species. So among the enterobacteria, we are most interested in Shigella and Salmonella. So highly selective media, they are more selective than what? Than selective media. So they select against many gram-negative bacteria also. Do you understand? right? So they allow only a narrow variety of microbes to grow. So they inhibit many gram-negative bacteria, and they are also differential. They differentiate bacteria according to pH change and H2S production. Now, from the previous table, we've seen that Proteus and Salmonella species are both H2S producers, right? Now, these two media, they cannot help us intelligence if this bacteria is an H2S producer or not, right? But highly selective media, in order to help us more in identifying Salmonella species, therefore, we include a system, some material in the preparation of the media to help us in seeing if it is an H2S producer or not. And H2S production mostly appears as what? As black discoloration, right? Black discoloration. When we see a colony with black discoloration, then this is what? This is a H2S producer, right? Now, these highly selective media, different highly selective media include Salmonella, Shigella agar, SS agar, pectine, enteric agar, HB agar, and xylose lysine deoxylate agar, XLD agar. Okay? We'll talk more in details about these media. Now, enrichment broths, only a problem. Okay, now, enrichment broths. Selenite and gram-negative broths are especially useful for encouraging the growth of Salmonella species while inhibiting other bacteria. So, if we suspect that this patient has Salmonella infection, right, then it is advised to put the stool specimen in what? In an enrichment broth before culturing it on solid media. Now, they are put in these broth, in one of these broths, and after hours of incubation, from 4 to 12 hours, depending, right, a sample is inoculated from this broth, from this tube of liquid media, onto a highly selected media, right? So if you suspect Salmonella, it is advised not to culture these two specimens directly on the agar, but to put it in a broth, incubate it for some hours, and then culture from it on a highly selected media, so that Salmonella will be encouraged to grow more and more. Selenite broth is better for retrieving Salmonella species, right? It encourages the growth of Salmonella species more, but the problem is that it kills Shigella, right? So if we are interested mostly in Salmonella, Selenite broth. If we want to retrieve both, if we are interested in both, we are not 100% sure, or we are interested also in Shigella, then we use gram-negative broth, okay? Now, McConkey agar, this is most likely a revision about McConkey agar, right? Now, McConkey agar, it is a selective bacteria for gram-negative bacteria, selective medium for gram-negative bacteria due to biosols and crystal virus. So it contains these two materials, and these inhibit the growth of what? Of gram-positive bacteria. Now, it differentiates bacteria that are able to grow on it according to lactose fermentation, right? So, it contains a pH indicator called neutral red. Now, it is advised because you will need several different pH indicators in this lecture, is that you arrange these information in the form of tables, for example. Right? I didn't arrange them in the form of tables because sometimes it is more useful for me and for you to discuss them likewise. Okay? But for your revision purposes, you can put them in a table form, right? After understanding what you mean by it. Now, neutral red, it is a pH indicator. Whenever we see a pH indicator, then we must know what color does it take in the acidic pH and in basic pH, right? So in acidic pH, it is red. In basic pH, it is yellow or colorless, okay? Yellow or colorless. It means that lactose fermenters, sugar fermentation in general, it will lead to what? To a drop in pH, increase in acidity. Right? So, lactose fermenters have a pink color, right? Because red discoloration, right? More close or closer to pink color. Lactose non-fermenters have colorless colonies, right? Now, this is the appearance of McConkey agar unnucleated. I've been in order for you to see what the baseline culture of the medium, right? So you can see it is pink translucent, the McConkey agar, right? Now, E. coli on McConkey agar, it gives, because E. coli ممكن انك تحاولوا شو اسمه تظبطوا من البروجيكتور شوي.
البروجيكتور عندك مش
البروجيكتور هون؟ Now, of course, I will be giving you these slides so you can see them more clearly on your computer screen. But still, here you can see that E. coli, it is a lactose fermenter, so they take the shape of what? Of pink colonies, right? Now there is something that we call Okay, now, I hope that the next pictures is clear, but the next pictures I hope will be very clear. Now, E. coli, lactose fermenter, right? It will drop the pH, it will cause pink discoloration, it will change into other color, therefore it will get pink colony. This is what we call bisalt precipitation. Bisalt precipitation, it takes the shape of what? Of more reddening outside the colonies, right? So not only the colonies will become pink, but also around the colonies there is an area, right, like a halo of more red discoloration. Now, Klebsiella species on McConkey agar. Klebsiella is also a lactose fermenter, right? But Klebsiella, it has mucoid colonies. You can see them, they are mucoid, right? Right? They are also lactose fermenter, but they do not show lactose or they do not show bisalt precipitation. The media in between the colonies is clear. Now, lactose non-fermenters on McConkey agar. Lactose non-fermenters, Shigella salmonella will take this shape, right? They are what? They are clear, right? They are clear, colorless, right? So lactose non-fermenters on McConkey agar. Now, E&B agar, it is similar to McConkey agar in that it is selected for gram-negative bacteria, but due to the presence of eosin and methylene blue, from its name, eosin and methylene blue. These inhibit the growth of gram-positive bacteria. It is also a differential media because it differentiates bacteria according to lactose fermentation. Now, eosin and methylene blue, they give a purple color and acidic pH, no color and basic pH, right? So, lactose fermentation, purple, right? And lactose non-fermenters will be what? Will be colorless. We'll see the pictures, okay? Now, E. coli, because we've said that E. coli is a very strong lactose fermenter, so the acidic pH will be what? The acidic pH will be more pronounced, right? The pH will be still lower. This means that the strong acidic environment will give what we call a green metallic sheen. Sheen, it is like a form of a shine. I'll show you. يعني زي كأنه في لمعة. Okay? هسه بنشوفها. It is a very characteristic appearance and helpful for us. Right? This means that strong lactose fermenters like E. coli have purple colonies with metallic sheen. Lactose fermenters, right, which only drop the pH mildly, they will cause only the appearance of purple discoloration. Lactose non-fermenters, they have colorless colonies because they will not drop the pH. Now, this is an uninsulated E&B agar, right? Its color is more reddish. It is also translucent, but it is more reddish than McConkey agar. Now, this is what we call the metallic sheen, right? When you see it, when you put it in your hand, you can see light reflecting on its surface, right? So, this is what we call a green metallic sheen, a shining green appearance, okay? Like phosphoric color, okay? Now, Klebsiella species on E&B agar, they are mucoid also, but they have a purple color, and lactose non-fermenters on EMB agar. There is no perfect agar. Now, SS agar. The properties of SS agar, they come from selection for Salmonella and Shigella due to bile salts and sodium citrate in high concentrations, right? So bile salts, when present in high concentrations, they will inhibit the growth of even some gram-negative bacteria. It also differentiates all highly selected media. They differentiate according to pH change and H2S production. Now, if we start with H2S production, it is the same among all three, right? So, differentiation according to H2S production, H2S producers show blackening of the colonies, non-H2S producers lack any blackening, right? So, whenever you see a blackening of the colony, there's a black center, it means that this is an H2S producer. It is Salmonella or Proteus. Salmonella or Proteus, right? Most likely. These are the ones that we are interested in at this level, right? Now, differentiation according to lactose fermentation. Now, the pH indicator that we use is the same as that of McConkey agar. So, it is fairly simple, right? It is neutral red. So, neutral red here, it is the pH indicator in SS agar. Lactose is the sole sugar. It means that if the bacteria is the lactose fermenter, it will drop the pH and neutral red will change to our pink or red color, right? If it is unable to ferment lactose, the pH remains high and neutral red remains what? Yellow or color, right? Now, this is SS agar. It is similar in appearance to McConkey agar, right? Now, this is E. coli on SS agar. It has the same what? It has the same appearance as that on McConkey agar, right? Because the same pH indicator, and E. coli, it cannot what? It cannot produce pH. It cannot produce, H2S. Therefore, there is no blackening of the colonies. But on McConkey agar, even if it is an H2S producer, it will not produce a black colony because there is no system in the medium to what? To detect H2S production. But here, E. coli, it cannot produce H2S from its own, right? Its physiology doesn't allow it to produce H2S. Therefore, it has the same appearance as that on McConkey agar, right? Even you can see here the bile salt precipitation more clear, right? You can see that in between the colonies, there is what? There is precipitation. There is a pink color. Now, Shigella species on SS agar, Shigella, it is a lactose non-fermenter. Therefore, the colonies remain yellow or colorless, right? Because neutral red, it remains in the basic, in the basic range, and it is it cannot produce H2S. Therefore, there is no There is no blackening of the colonies. Now, Salmonella, it is different, right? Now, Salmonella, it does not produce or it does not ferment lactose. Therefore, the colonies are what? Are colorless. But because it is an H2S producer, you can see the The dots on the or in the center, right? You can see them clearly. It means that it is an H2S producer, right? Now, HE agar. HE agar, like SS agar, it is a highly selective medium. It means that it selects against many gram-negative bacteria because of the presence of bile salts in high concentrations, right? So it gives more space for Salmonella and Shigella to grow, right? Now, it differentiates bacteria according to sugar fermentation and H2S production. H2S production, the same principle as that of SS agar. H2S production, black discoloration. No H2S production, no black discoloration. Right? Simple enough. Now, differentiation according to sugar fermentation, here the pH indicator is different. Right? The pH indicator is different. It is called thymol blue. Thymol blue. Now, thymol blue, it is a pH indicator. It is yellow in acidic pH and blue in basic pH. Right? Yellow in acidic and blue in basic. Now, here, the sugars present in HE agar are what? Are lactose and sucrose. Lactose and sucrose. It means that if the bacteria is able to ferment at least one of these, either lactose or sucrose, then the color of the column would be what? Would be orange to pink, right? Orange to pink. Because of the yellow color mixing with the blue background, it will give us an orange to pink color, right? So, an orange to pink color, it indicates that the bacteria is able to ferment at least one of what? Of the sugars, right? Now, if the bacteria is unable to ferment either one of these, then they will what? They will take the color of the background. The color of the background is blue. I'll be showing you the medium now, okay? So, Salmonella and Shigella are known to be unable to ferment neither lactose nor sucrose, right? So, Salmonella and Shigella will have what? Colorless blue colonies. Now, Shigella, colorless blue colonies with no black discoloration because it cannot produce H2S, but Salmonella, the blue colony with a black center, right? Now, this is an uninflated HE agar. You can see that it is a blue-green medium color. Sorry, a medium which is blue-green in color, right? Now, this is E. coli on HE agar. Now, this is what? An uninflated part of the plate, right? You can see it still has the what? The green color. But here, the growth of E. coli, it has caused a drop in pH, and so thymus blue has become what? Yellow, right? And so mixing of the yellow color with the green background, it has given us what? This yellowish orange discoloration, right? So this indicates that it can, what? It can ferment either lactose or sucrose, right? So we're sure that this is not Salmonella or Shigella, right? Now, Shigella, it grows on HE agar, but it cannot produce what? A drop in pH. Therefore, they do not cause a change in the color of the background, right? And they have what? They show no black discoloration because they cannot produce H2S. Salmonella species, right? You can see clearly the black discoloration, but if you concentrate, you can see that there is no yellowish discoloration. It means that this bacteria produces H2S, but cannot ferment neither one of these. Cannot ferment lactose and cannot ferment sucrose, right? So this is characteristic of what? Of Salmonella, right? Now, finally, there is XLD agar. XLD agar. From the scheme, X xylose, which is a type of sugar, L lysine. So xylose here, lysine here, and deoxycholate. Deoxycholate are the what? Deoxycholic acid, the bile salts, right, that we use in this medium. So, XLD agar, it is also a highly selective medium, right? It selects against many gram-negative bacteria as well as gram-positive bacteria because of the bile salts. It differentiates according to H2S production because it has an H2S detection system. Now, it differentiates according to sugar fermentation. Sugar fermentation. Now, the pH indicator that we use here is what is called phenol red. Phenol red. You've met phenol red in regular iron agar and triple sugar iron agar. Okay? We'll be revising these in a moment. Now, phenol red, it has the opposite color change of what? Of neutral red. Neutral red, it was red in acidic pH and yellow or colorless in basic pH, right? But phenol red, it has the what? It has the opposite, what? The opposite color change. So, in acidic pH, it is yellow. In basic pH, it is what? It is red, right? So, the opposite, right? Now, here we have how many sugars? In SS agar, we only had lactose. In HE agar, we have lactose and sucrose. In XLD, we have also X xylose, right? So, lactose, sucrose, ferment either sugar tend to give yellow colonies, right? Understood? Now, E. coli can ferment lactose, so it produces a yellow color. Shigella can ferment none of these, therefore it gives colorless colonies, right? Now Salmonella. Salmonella, it is what? The story has more than one element. Salmonella can ferment xylose, therefore at the start of the incubation, it gives a yellow color. But because this medium contains the amino acid lysine, then it starts a process called lysine decarboxylation. You know, lysine is an amino acid. It has a carboxyl group and an amino group. Now, when Salmonella removes the carboxyl group of this amino acid, it will increase the what? The pH. Therefore, it starts as a yellow colony during incubation, but at the end of the incubation, it will what? It will shift the pH from the acidic more towards the basic. Do you understand? So initially, it starts as yellow, but after a while, right, we incubate it for 18 to 24 hours, right? So there is a long time. After a few hours, it starts as a yellow colony, but after that, lysine decarboxylation, it will make it what? A red colony, right? So it will change the pH and the color of phenol red back towards the what? The basic spectrum. Now, this is an uninoculated XLT agar, somewhat like that agar, right? Red in color. Now, this is E. coli, right? E. coli, it produces a yellow color because it changes phenol red, the color of phenol red, towards the what? The yellow color, right? So, yellow color, you can compare it with the uninoculated part of the medium here, which is what? Redder. Now, Shigella, it can ferment what? It can ferment neither sugar of those present in the medium. Therefore, it remains what? It remains clear, right? And there is, of course, no black discoloration. Now, Salmonella, you can see that there is a black center, right? And there is on the periphery a red color, right? The red color is not due to sugar fermentation or the inability to ferment sugar, but due to the what? Lysine decarboxylation, right? So it changes it back to the what? To the basic color. Now, this is a very fast revision of what? Of biochemical reactions that you've meant. Indole production. This is an uninoculated tube. If it is unable to break down indole, right, or break down tryptophan, then it will what? It will remain colorless on the surface. But if it produces tryptophanase, then it will what? It will give us this deep red layer on the surface of the medium. Methyl red rest. This is an MRVP. MRVP meaning what? Methyl red bogus cross cover drop, because they are integrated in the same drop. The bacteria to be tested against methyl red and bogus cross cover are integrated in the same drop. If it is negative, it remains yellow colorless. If it is positive, it becomes what? Red, right? So this is a positive reaction. Bogus cross cover, unintegrated, negative, yellow, positive, pink to red. Here it is a deep red color. Usually it is pinker in color, bogus cross cover. And citrate agar. Citrate agar, it takes the form of a slant put in a tube, right? This is an unintegrated medium, negative, remains green, positive because it changes the color to the basic spectrum, therefore it becomes positive. This is urea agar, right? Urea agar, unintegrated, negative, and positive. Positive, because of the production of urease, it breaks down urea into what? Into ammonia, and ammonia increases the pH and changes the color of this pH indicator. What is this pH indicator? Phenol red, the same as that in XLT, and triple sugar iron agar, and in KRA, regular iron agar. Now, this is regular iron agar or TSI, triple sugar iron agar. We are not sure, okay? Now, if you see a medium put in a slant in a tube like this, you have to ask whether it is regular iron or triple sugar iron. Now, what is the difference between regular iron and triple sugar iron? Do you remember? Do you want me to explain these in a hurry? Or do you know them?
No.
فاهمينهم ولا لا؟
ناسينهم.
لا مش فاهمينهم.
ناسينهم.
ماشي. Okay. Now,
كان البرودكشن تاع الجلوكوز.
regular iron agar and triple sugar iron agar. Starting with the simpler one, which is regular iron agar. Okay? Now, starting with regular iron agar. Okay, you can understand regular iron agar. Of course, it is a solid medium. It is an agar, but it is put in a test tube, right? In a test tube in the form of a slant. You know what is a slant,
yes.
right?
Ma'il.
Okay,
yes.
You know what is a slant,
yes.
right?
Yes.
It is not a deep. Now, here you have to understand that these tubes, they give us two forms of environments. The slant part, or the top, and the bottom.
Aerobic and anaerobic.
Now, the top, it provides us with an aerobic environment, with oxygen, exposure to oxygen.
Yes.
The bottom part, because it is covered by more media, therefore it is what? It is anaerobic. There is no oxygen production. Now, regular iron agar, you have to understand it as being made of two systems. It contains materials which belong to two systems, right? One of them is for sugar fermentation, a pH system, and the other is an H2S system, right? H2S system similar to those that we have discussed. An H2S system, it will lead to blackening, right? Black discoloration. So put it aside for now. Let us now concentrate on the pH system, okay? Now, the pH system, here the pH indicator that we use is phenol red, right? Phenol red, it is red in basic pH and it is yellow in acidic pH, right? Now, what are the processes that change the pH of the medium?
Fermentation.
What are the processes that change the pH of the medium? There are two processes. One of them is called sugar fermentation, right? Sugar fermentation, it changes the color of the medium towards the what? Towards the yellow color, right? Yellow direction. The other process is not sugar fermentation, is what we call peptone oxidation. Oxidation of what? Of peptones. Peptones are protein degradation compounds, right? So we have a sugar fermentation process, right? It changes the color towards yellow, and there is a peptone oxidation process. It changes the color towards what? Towards red, okay? Because it leads to a basic pH. It changes the color of phenol red to the opposite direction. Now, you have to understand there is a very important what? Very important point to understand here. Is that what? Sugar fermentation, it takes place where? It takes place in the top and in the bottom. Because it is oxygen independent, yes. Right? So, sugar fermentation, it happens here and it happens here. Peptone oxidation, it only happens where?
In the top.
Okay? It does not happen in the bottom. It is an oxidation process, it needs oxygen. Right? Now, what are the types of sugars that are present in KIA, in regular iron agar? There are two sugars that are present. One is glucose, the other is what? Lactose. Okay? Now, the difference here, you have to remember this, is that glucose is present in relatively small concentrations. So it is consumed rapidly. Lactose is present in a very high concentration, so it needs more time to what? To get consumed. Okay? Now we can start by predicting what are the different possible KIA appearances and different what? And different cases of sugar fermentation. Right? Now, let us start. Suppose that the bacteria is originally, of course, the medium is what? It is titrated in a red color. You have to start, you have to remember that. We start with a red color. We titrate the pH so that the phenol red is what? Phenol red is more in the red color rather than the yellow color. So we start with a somewhat basic pH. Now, suppose that this bacterium is a glucose non-fermenter, a lactose non-fermenter. It cannot ferment neither glucose nor lactose. Right? Now, what will happen?
Nothing.
The bacteria, we inoculate it in the medium and we incubate it. It will find glucose and lactose, but it cannot ferment neither. Therefore, there is nothing that will drive the pH towards the what? Towards the yellow direction. Right? Even after a while, peptide oxidation will start, and this will drive, especially the upper part of the tube, towards more and more more reddening. Right? Therefore, after incubation for 20 to 24 hours, the tube will be what? Will be red all over, right? In the top and in the bottom. The results of KIA, we write it as a fraction, right? Here, K over K means alkaline over alkaline. It means red over red. Do you understand that? Yes. So if it is red all over, then it is what? Red all over. It means that it is a glucose non-fermenter, lactose non-fermenter, no fermentation. Okay? Now, the second possibility is what? It is a glucose fermenter. For now, just remember that it is a glucose fermenter, but what? We will not ask about lactose for now. Now, glucose fermenter, we incubate the medium after inoculating it with the bacteria. Now, it will start growing. It will find glucose. Very good, okay? So, it will find glucose in the upper part and in the lower part. Fermentation will take part in the whole medium, will take place in the whole medium. Therefore, the whole medium will shift towards the what? The yellow color. Do you understand? Right? Now, the problem here is that glucose is present in only small concentrations. So, it is consumed rapidly. Right? It will not last for the whole incubation period of 20 to 24 hours. Right? It gets consumed in about what? 10 to 12 hours. Right? Do you understand? So, if we open the incubator after 10 to 12 hours before the incubation period is over, we can see that the tube is yellow all over, then we call it what? We can say that it is a glucose fermenter. Now, after glucose gets consumed, there will be no more drop in pH unless the bacteria is what? Is a lactose fermenter. Right? So, suppose here that the bacteria is a lactose fermenter, then it will find plenty of lactose to what? To ferment. Therefore, the pH will keep on dropping and dropping and dropping. And so the yellow color will deepen. Right?
Deep, yes.
Peptide oxidation will try to change the color of the upper part of the tube back to the red, to the red direction, but it will not be able because lactose fermentation will be very pronounced. Right? So peptide oxidation will not be able to change the color of the top back to the red direction. So if we open the incubator after 20 to 24 hours and we see that the whole tube is yellow, then we say it is a glucose fermenter and a lactose fermenter. Okay? Now suppose that it is a glucose fermenter but lactose non-fermenter. In this case, after 10 to 12 hours, the whole tube is yellow. After that, the bacteria is unable to ferment lactose. Here, peptide oxidation will come, and it will change only that color of the top part of the tube back to the red direction, because peptide oxidation cannot take place in the bottom part. Therefore, if we see after 20 to 24 hours that it is red in the top but yellow in the bottom, we can say that it is a glucose fermenter, lactose fermenter. Do you understand it? Okay? So this is the idea or the principle behind KIA. Of course, if we can see any black precipitate, right, then it means that it is an H2S producer. Now, suppose that the upper part of the tube is red and the whole lower part is black. Then we can say that it is what?
Glucose fermenter.
It is a glucose fermenter.
Non-lactose fermenter.
Lactose fermenter why?
No, non-lactose fermenter.
Why non-lactose fermenter?
لأنه صار red.
Okay, so it is a glucose fermenter, lactose fermenter, and what H2S producer. Okay, now suppose that the whole tube is black.
معناها
يكون ال H2S.
How do you know what color is beneath the black color? If it is red or yellow? Usually, if the black color covers the whole or half of the tube, then we can say that it is what? It is yellow beneath. Why? Because H2S production, it needs hydrogen ions, it needs what? It needs acidic environment. Right? So it needs a yellow color. Okay? So in principle. In some cases, it is red at the top, right? It is yellow at the bottom, and there is a black ring in between. This is also K over A with H2S production, right? So it is a glucose fermenter, lactose non-fermenter with H2S production. Okay? Now, TSI agar from its name, this is triple sugar iron agar. Of course, the iron here, it is related to what? To H2S, it is an H2S indicator. It is related to the H2S system, you don't have to concentrate on it. Okay? Now, triple sugar, what does triple mean? It contains glucose in low concentration, but lactose and sucrose in high concentrations. It means that if the bacteria is able to ferment either sucrose or lactose, then the what? The whole tube will remain yellow. You understand? Right? We've said that Salmonella and Shigella are unable to ferment neither this nor this. Therefore, in TSI agar, if we see that it is K over A reaction, it means most likely that it is Shigella or Salmonella. Right? If it is Salmonella, of course, there will be some degree of that discoloration. Do you understand? Okay? So triple sugar iron agar, I have to tell you that this is a KIA or TSI. If it is TSI, it means that what? It means that it contains lactose and sucrose. It means that if the whole tube is yellow, it is glucose fermenter, lactose or sucrose fermenter, fermenting either at least one of them. Right? If it is unable to ferment neither sucrose nor lactose, therefore it becomes K over A. Right? Now, Enterobacteriaceae, by definition, are glucose fermenters. Therefore, they will always give us what? A yellow part. Right? They will always give us a yellow lower part of the tube. Right? Glucose fermenters, all Enterobacteriaceae. Pseudomonas aeruginosa is unable to ferment glucose. Therefore, it will give us a K over K reaction. Now, E. coli, it is a glucose fermenter, lactose fermenter, non-H2S producer. Right? So it will give us what? A over A. Right? Now, Shigella, Incligular iron agar, and TSI, it will give us both K over A. Right? It will give us K over A. Red, slant, and yellow, bar. Now suppose that the bacteria has given us a K over A reaction in KIA and A over A reaction in TSI. It means what? It means that it is a glucose fermenter, non-lactose fermenter, but sucrose fermenter. Right? Now, this is Salmonella. You can see K over A with H2S production. Right? Now, other important bacteria, enterohemorrhagic E. coli is mostly of the serotype O157H7, right? Now, it is biochemically differentiated from other E. coli strains by being a sorbitol non-fermenter. Sorbitol non-fermenter. There is a sugar called sorbitol. Most E. coli strains are able to ferment sorbitol except for this part, for this strain, PHEC. So it is a lactose fermenter, but sorbitol non-fermenter. Now, therefore, we have a medium called sorbitol McConkey. Sorbitol McConkey, the same principle behind McConkey. But the difference is that instead of lactose, we add sorbitol. Therefore, if it is a sorbitol fermenter, it will give us what? A pink colony. If it is a non-sorbitol fermenter, a clear colony. Now, this is an uninucleated sorbitol McConkey agar, similar to McConkey agar in appearance. Now, here you can see two different types of colonies. The first one is the pink colony of E. coli, right? You can see some white precipitation here. And the one indicated by the arrow is a colorless colony, right? This colorless colony is what is E. coli 0157H7. Why? Because it cannot ferment solvitor, right? Now, Campylobacter jejuni can grow on routine non-selective media such as blood agar and McConkey agar. But we inoculate what different, there are selective media, especially for it. CAMP-TBAP, blood agar plates, Campylobacter blood agar plates, Scleromedium and Bacillus medium. It is microaerophilic, it needs to be incubated in 5 to 10% oxygen, right? Capnophilic, it needs a high concentration of CO2, and thermophilic, therefore it needs 42 degrees Celsius. Above that, 37 degrees Celsius, we usually incubate bacteria in. The media are incubated for up to 72 hours, right? So we incubate it for up to 3 days. Each day we look for broth. If there is no broth, then we can say that there is no Campylobacter jejuni in this sample. Campylobacter jejuni is a curved gram-negative rod, so it is not straight, right? It is oxidase and catalase positive. Okay, this is E. coli 0157H7 on solvitor McConkey agar. It is colorless. It does not have the pink color of other E. coli strains. Now, this is a gram stain of Campylobacter jejuni. It has what we call a seagull appearance, right? Because it is curved. It has a spiral shape, right? So you can see like a seagull, like this, okay? This is what we call a seagull appearance. Seagull appearance, okay? Like seagulls seen from the horizon. Now, Vibrio cholera, it is an oxidase positive curved gram-negative rod. It is also curved. It has a comma-like shape. We have an agar selected for Vibrio species called thiosulfate citrate bile salt sucrose agar. Okay? Now, from its name, bile salts, it makes it what? A selective medium. It inhibits against many other gram-negative bacteria. Right? Thiosulfate, just ignore it. Citrate and bile salts, they are inhibitors, so they make the medium what? They make the medium selective. Right? Thiosulfate, ignore it, because there is an H2S system, but we will not talk about it here. And sucrose. Please concentrate on sucrose. Okay? Now, bromothymol blue is the pH indicator. Like thymol blue, it is yellow in acidic pH, but blue in basic pH. Bromothymol blue is not the same as thymol blue. They have other different characteristics. But they have the same color changes. In the acidic range, it is yellow. In the basic range, it is what? Blue. Now, if the bacteria is able to ferment sucrose, then it will change the color to what? To yellow, like Vibrio cholera. And other non-fermenters, there are some Vibrio species which cannot ferment sucrose, therefore they will give what? Bluish colonies. This is a TCBS agar, it is green. This is mountain here. Okay, now this is on TCBS agar, Vibrio cholera, yellow colonies because of sucrose fermentation, right? It will change the color of bromothymol blue towards the yellow range. And non-sucrose or sucrose non-fermented on TCBS agar. Okay? This is a gram stain of Vibrio cholera. You can see that it is what? It is curved, like a cone, right? It is curved. Now, basic principles of GI bacteriology lab, we have only 2 slides. Appearance of the fecal specimen gives an idea about the possible cause. Shigella, Campylobacter jejuni, and enterohemorrhagic Escherichia coli. So we observe the stool specimen. If it is an unformed stool, right, towards the liquid state more, with post mucus and blood, then we suspect that it is caused by Shigella, Campylobacter, or EHEC, E. coli 0157H7. Salmonella species, unformed or watery stool, less chance of post mucus and blood. Okay? Vibrio cholera, it doesn't contain pus or blood, but it contains what we call rice water stool, because watery diarrhea with mucus flicks, okay? Routine culture is done on a selected medium, such as McConkey or E&B, highly selected medium, such as SS agar, HE agar, or XLD agar, and a medium for Campylobacter. Okay? So 3 plates, right? Prior enrichment in broth, selenite broth or gummy broth is to be considered, right? Especially if we suspect that it is Salmonella. Culturing for other organisms, such as sorbitol McConkey for E. coli O157H7 or TCBS for Vibrio species, is only done upon suspicion, right? Suspicion from the clinician, من الدكتور, okay, from the treating physician, or suspicion from the observation of the fecal specimen, right? But these are the 3 that are what? That are routine. Serology plays an important role for such organisms as Shigella, Salmonella, EHEC, and Vibrio cholera. Okay? So serology is important also here. Right? O157H7, it means that it is a strain differentiated from other E. coli strains by what? By serology. Right? So serology is important here, very important for Salmonella, very important for Shigella, and for Vibrio cholera. We're supposed to have learned about that in the theory lectures. Toxin assays for toxin-mediated diseases like Clostridium difficile or pseudomembrane enterocolitis, and food poisoning, Staph aureus, Bacillus cereus, etc. are also available. Okay? Yeah. Okay. Now, 10 minutes, then we will restart the parasites. Okay?